I'll quickly introduce Marisa. She's an assistant professor at the Columbia University Irving School of Medicine, and I'm very, very happy she took the trip over here from New York, and thank you so much for being alert, for giving us this now. She does very exciting stuff.
Is it just this button? Okay. Thank you so much for having me today, and very excited to share some of my work with you. And ooh, wait, this way. So I'm just pressing this. Oh, easier than I thought. Okay, great. We've talked a bit about inflammation actually today already. So I'm interested in maternal immune activation. I will say currently right now I'm studying healthy pregnant women and looking at sort of variation in immune function. To then sort of get to a next stage where I can do kind of comparisons. But the reason I think maternal immune activation is particularly interesting is because it's a very sensitive system, particularly during pregnancy. And so what I'm talking about when I say that is really it could be sort of any activation of either the innate or adaptive immune system. It's really sort of just could be global inflammation in response to either external or internal stimuli. And again, it's very sensitive, so we know that you can have elevations in inflammatory markers due to infections, poor physical health, mental health and stress as well, and environmental insults. And so really in terms of pregnancy, you're gonna see a lot of variation, and within pregnancy in and of itself, right, you're getting a lot of variation in the immune system over the course of the pregnancy. And so, so what is currently known, really epidemiologic studies were really driving our understanding of how maternal immune activation was associated with particularly psychiatric risk or behavioral risk in humans in particular. And so there are a number of seminal studies that identified that maternal infection was associated with actually several psychiatric disorders, first with schizophrenia and then more recently autism and bipolar disorder. And then in terms of clinical studies, particularly preterm models, really led the way in starting to understand, okay, in humans, yes, there are associations with the brain, particularly early in development. And, you know, so it's something that we should be considering and thinking of. But sort of more kind of later time points, you know, there's some head circumference work, particularly with preterms, that was also providing information in terms of the human specifically. And then I think we all know that there's a really robust literature in terms of rodent non-human primate models, and that's really what led the way in terms of really sort of taking a deeper dive and understanding what particular brain regions and systems might be particularly vulnerable to maternal immune activation or inflammation. And so some examples from that literature were the prefrontal cortex. So, you know, we are thinking about cognition, executive functions, hippocampus, and parietal regions. And then the decrements in behavior that were shown were related to memory and behavioral reactivity and inhibition, which I'm particularly interested in, as well as attention. And so the gap, again, sort of, you know, at that time when I started this work was, okay, what about the human brain? What about the early human brain? And so, so what my lab does is, it's the early neuroimaging, neuroimmune, neuropsychology lab, and I'm really focused on understanding how maternal immune activation inflammation during pregnancy is associated with early brain development, and then later how that relates to psychiatric risks, but early in development. And so here what I'm going to show you today is some of the work that I've done looking at multimodal imaging and how particularly a number of pro-inflammatory cytokines with some of the early work I was doing are related to different imaging modalities that you've actually heard about today already. So that's good. So thank you everyone else. But focusing on these three: functional connectivity, structure, as well as within the context of structure, diffuse and tensor imaging, and then MRS as well. And then, so broadly, we actually have at Columbia kind of a number of people doing this work where we've been able to kind of pool our data. And so, so kind of broadly, there's around 200 women that I was able to utilize for these, the subsets of these studies, starting in teen years, so age 14, going up to age 40. And again, in the context of this early work, it's really healthy pregnancies. I'm starting now to look at women with chronic or acute health conditions. And these women underwent blood draws in the second trimester and third trimester. And originally, only IL-6 and CRP were actually measured. Now I have a much more robust immune panel, but the work I'm going to show today is going to be related to that. And the reason that those were selected by some of the original investigators is because, particularly IL-6, had a strong association with psychiatric risk. And as we are talking about, newborns, neonates, so these kiddos underwent MRI between 2 and 6 weeks gestation, and it's unsedated. And I do daytime scanning, which is, I think, a bit unique, so I just wanted to kind of acknowledge it. So it's unsedated during the day, I don't do any nighttime scanning. And actually it was because my scanner wouldn't allow that, and so you have to work with what you have. So I just wanted to mention that. All right, and we have a GE scanner. Okay, so our first paper in 2018, and actually that year, 2 other papers came out by one other group, where we focused on the third trimester, and we used both immune markers and looked to see actually if they were related to not only postnatal outcomes, newborn MRI, and toddler cognition. So we had the Baileys, but we also looked at fetal heart rate as an index of kind of autonomic development and kind of precursor early kind of regulatory processes. And we focused on specific seed regions, again, just trying to kind of, essentially, it was a smaller sample size, and so we didn't want to do start with whole brain analyses, and also, again, I'm particularly interested in regulatory brain processes, and so we focused on the salience network, which is composed of the dorsal anterior cingulate as well as the insula. And again, it's involved in regulatory processes, behavioral, and also is also integrated with sort of early autonomic development as well. So it sort of was a nice kind of complement to that other prenatal data that we have. Okay, so as I mentioned, so we did seed connectivity, and so what I'm going to show today is just the findings with the dorsal anterior cingulate, so not the insula findings, but that's in the paper, and I'm happy to talk more about that later. And so again, we're looking at functional resting state connectivity, so the baby's just naturally sleeping, and we're just essentially taking that seed region in relation to like any other region in the brain, and we're looking at kind of time course correlation essentially, and again focusing on the DACC. And so for IL-6, what we found was for higher levels of IL-6 in that third trimester, there was an association with medial prefrontal cortex, so it was reduced connectivity. And then we actually see a similar pattern with higher levels of CRP in terms of reduced connectivity in the prefrontal area. Then in addition, there's significance with the basal ganglia when we're thinking about motor coordination, the cuneus occipital lobe, and temporoparietal junction. And so we had sort of more robust findings with CRP. And then kind of just to sort of pull this all together, if you look kind of across the two immune markers, these were sort of the main findings: medial prefrontal, basal ganglia, and temporoparietal junction. And at that time, one of the things that we were thinking is that we have these brain regions with the salience network, because like I mentioned, we also looked at the insula, and some of those brain regions that were also significantly connected were part of these other two networks. So the central executive and the attention network default mode. And so we were thinking that there could be something interesting about this sort of tri-network that's happening in relation to the inflammation and its relationship to early brain development, because the salience network really functions as sort of the switching mechanism between these two other networks. So that was kind of exciting, and a lot of the work we do and grants we submit now really does sort of hang on those sort of brain regions as the primary area of interest. Although we obviously do whole brain analyses as well as our sample size gets larger. And then just to think about the behavioral associations, so we did find that with increased CRP, there was an association with reduced fetal heart rate variability. And then in the third trimester, your expectation is that you're going to actually have increased fetal heart rate variability. So it would sort of be going in the opposing direction. And then in terms of our toddler outcome, cognition from the Bayley scale, there was actually increased or higher scores on that cognitive measure in association with both the immune markers, which again, we see that happen as well too. People have talked about this as potentially sort of accelerated growth, and I know that all of us that do this early work too are also cautious about sort of how we interpret or over-interpret our results, right? Because this is a really rapid period of growth, but that is what the finding was. It's a small sample, and obviously should be replicated. And then on to these other imaging modalities, because obviously that's not the whole story, just functional resting state data. And so at that time, there was really only one other study that was looking at microstructural organization using fractional anisotropy, so DTI, again, we've talked about today. And they really focused on one particular track, the uncinate, to kind of be thinking about, again, these more limbic processes. And at that time, there was no investigation specific to brain metabolites. So for this one, we actually didn't have sort of a particular area or tract that we were considering. We also had a larger sample size, and we've already talked about what fractional anisotropy is measuring, so I'll skip over that. And you have the results with CRP and IL-6 side by side now, and really kind of the thing that stands out is, with higher values of the immune markers, for CRP, you're seeing a decrease, but also primarily in frontal regions, and for IL-6, it's more of these subcortical regions in the thalamus. And then just to kind of, so our approach for MRS at this time point was a whole brain approach. And so these are the metabolites that we're able to reliably obtain, and they're also really sort of broad indices of brain development. And so again, for us, it was a good start to start with these. And so first, just the IL-6 results, and what really stood out to us is really that regardless of which of the metabolites you're looking at, is really similar pattern of results. And And then similarly for CRP, you see a really, regardless of whether sort of there's increases or decreases, it's a very similar pattern of results sort of across the different metabolites, which, you know, could be particularly interesting and useful when we're thinking about sort of clinical targets. And this kind of, just to sort of recap those two pieces. So some of the main findings were, that seemed common in terms of looking at fractional anisotropy, was the anterior temporal lobe, but otherwise, as you saw, like really there was a lot of sort of differences between the two immune markers. And then in terms of the brain metabolites, there seemed to be some consistency around the insula and basal ganglia. But again, you know, some divergent results, really suggesting that, you know, the multimodal approach, I think, is pretty critical when we're thinking about inflammation. And then, as I said, kind of, you know, I have a number of other cohorts that we're collecting data on. We have a much more rich immune profile, and there's other things that, you know, I'm considering too. And so I just wanted to kind of share some of those results. These are, you know, unpublished right now, but one of the things that I was particularly interested in is the timing of immune exposure, because some people in the field, particularly that are doing human work, they might just sort of average values over pregnancy, but then there are other people that feel very strongly, no, it should be sort of within a discrete time window. It shouldn't be averaged over pregnancy because we know there's variation in immune function during pregnancy, right? So, you know, we were interested in identifying, okay, if we take our second trimester and third trimester data, you know, are the effects on the brain similar? And so we had some, this is the DTI data and the MRS data, and we just compared those, looked at differences. And as you can see, in terms of whether sort of it's a pattern of increase in relation to the immune marker or decrease, there's really clear similarity between the two trimesters, and there's, you know, very few that would be significantly different. So really it's the thalamus that's under 0.05, and again, this is a lot of comparisons, and the cingulate for MRS. So maybe it is okay to average, you know. Again, more work still needs to be done, but, you know, we just wanted to start thinking about that and put that data sort of side by side. And then the other piece that I'm particularly interested in, and I've been doing longitudinal scanning within the first year of life and now fetal scanning, is to understand what are the brain changes that are associated with exposure to maternal inflammation. And then, and so here's just an example of that work. I have some things that because of time I'm not able to show, but again, happy to talk more. And so this is kind of going back to the MRS data, and the way that we're now collecting MRS data is actually using S-laser, and we're doing kind of ROI analysis now instead of whole brain, so we can essentially quantify more metabolites. I was particularly interested in myoinositol because that is the metabolite that's been most associated with inflammation, and there's actually now some treatments that you know, particularly target these systems. So I was particularly interested in this. And so what we did is, in, you know, a set of infants that I've been longitudinally scanning, we were able to quantify this over time across different brain regions. So we have the anterior cingulate, the cerebellum, the occipital lobe, prefrontal cortex, and temporal-parietal junction. And you can see that in earlier periods, you have higher levels of myoinositol, and then, you know, that sort of shifts over the first year of life. What's interesting about this is when we're thinking about an inflammation indicator, myo-inositol, you'd expect to be higher. So, and again, we also know that a lot of things can be reversed in early development and then sort of shift back around school age. So, so that was an interesting finding for us. And again, we're continuing to do this work, not just with myo-inositol, but I'm particularly interested in this. I have collaborators who are interested in other metabolites. All right, so just kind of overall summary. So it seems like it was interesting to me when I kind of started this work that there really were very few studies in humans looking at the relationship between maternal immune activation and early brain development, particularly because there was such a robust preclinical literature. And so I'm glad to be part of that group that's doing that work now. Obviously, it's really important for development. It's particularly important for psychiatric risk, and I am in a psychiatry department, so that matters for us. And the findings also highlight that not only multiple measures of MIA are important. So again, this early work, we only had 2 markers, and I saw within that, okay, there are differences that are important. And I knew we needed to have, a more robust profile, which we do now. And also, I think multimodal imaging is really critical as well. And then just want to thank some particular individuals who have shared their data, and we pool our data, and our funders, and thank all of you for listening.